All right, I wanted to make a virtual lecture on one of my favorite topics we learn about in microbiology, and that's selective and differential media. Now, when we talk about selective and differential media, we have to take into account 2 points here, the selective part and the differential part. So let's define each. Selective media is one that inhibits the growth of certain organisms while encouraging others to grow. Now, this is actually very important because oftentimes when you're in a clinical setting as a clinical microbiologist, you often have what we would call mixed cultures. What that means is you have many different types of bacteria growing in one culture, or present, if you will, in one culture, and we want to only isolate those organisms which we're interested in. Therefore, selective media allows us to separate organisms based on, for example, their cell walls. So it's going to allow us to separate gram-positives from gram-negatives. When we talk about gram-positives, gram-negatives in a clinical setting, typically we're talking about gram-positive cocci from those gram-negative rods, right? The staph from the enterics, or the strep from the enterics. Now, differential media, this is the second part of it. When you have a mixed culture, you often have many different gram-negatives in that culture, for example. So what a differential media allows you to do is differentiate or distinguish amongst those organisms that have the ability to grow on that particular media. Right? And as we talked about before, we often use these media to isolate those pathogenic, those bacteria that make you sick. So let's give you 3 different examples of what I think are classic selective and differential media. First, mannitol salt agar. We're going to use this one in the laboratory. There's both a selective part and a differential part to mannitol salt agar. Let's first talk about what the selective agent in this plate is. Within the agar, we've set up 7.5% NaCl. We've set up high salt conditions. Now, as we learned, what are the names of organisms that can grow in high salts? That's right. Organisms that can grow in high salts are considered halophiles. Now, the reason this is advantageous is we talked about isolating gram-positive cocci from gram-negative bacilli. As we know, gram-negative bacilli are not halophiles, therefore they cannot grow on mannitol salt agar, while members of the genus Staphylococcus can, because Staphylococcus is considered a facultative halophile. Alright, so now you can imagine we've cultured the plate. Any gram-negatives that were present in that clinical specimen have been inhibited. The only things growing on that plate are those members of Staphylococcus. Now, one thing we have to realize is amongst these genera or genus of Staphylococcus, there are many different species, some which are pathogenic, some which make you sick, others that do not make you sick. So how can we differentiate members of Staphylococcus? We use a differential agent called mannitol, which is a fermentable sugar. As we learned about in lecture, fermentation is typically an anaerobic process in which we allow ATP to be generated through glycolysis. In the process of fermentation, we often have what 2 byproducts? That's right. We can often produce acids and also carbon dioxide as a result of fermentation. So, for those members of Staphylococcus that can ferment mannitol, acids will be produced, therefore the pH of the media will become acidic. However, for members of Staphylococcus that cannot ferment mannitol, the pH of the media should not change. So let's look here at the picture on the right. Here we see a member of Staphylococcus, Staphylococcus aureus, classic example. We see that media has turned yellow. That's an indication that we have acidic conditions within the media. Therefore, it's an indication that Staphylococcus aureus can ferment mannitol. On the right here, we have Staphylococcus epidermidis. We can see there is no change in the color. Therefore, no acid was produced because there was no fermentation. So that is differential. We see both members of staph grew, yet they look very different on a plate. Now, this actually is beneficial in being able to identify pathogenic Staphylococcus, because typically pathogenic members of this genus can ferment mannitol, whereas non-fermenters of mannitol are typically non-pathogenic. So when I look at this plate, what you see is Staphylococcus aureus is a pathogenic member of staph, where Staph epidermidis is a non-pathogenic member. So the beauty of the MSA plate, first of all, is we've inhibited gram-negative bacilli. So anything that grows we know belongs to the genus Staphylococcus. Next, we try to differentiate amongst members of Staphylococcus based on their ability to ferment mannitol. As we can see, Staphylococcus aureus can ferment mannitol, creating acidic conditions, whereas Staphylococcus epidermidis cannot ferment, therefore you see no change, no acid produced. Now let's move on to the next selective and differential media. Eosine methylene blue agar, another differential and selective media we use in the laboratory. Typically, we use EMB to isolate gram-negative bacilli. To be more specific, members of those enteric family. So let's talk about the selective and differential agents. The selective agent in the plate are the two dyes, eosin and methylene blue. Eosin and methylene blue inhibit gram-positive growth while allowing for gram-negative bacilli. So again, in a clinical sample, we're able to isolate just the gram-negative bacilli. Now, once we isolated the gram-negative bacilli, how can we differentiate amongst those members that grew? What is the differential agent? In this case, the differential agent is lactose. Lactose is a disaccharide that is fermentable. There are certain members of enterics that can ferment this particular sugar, other members that cannot. Now, because of the dyes that are the selective agents, those dyes also turn a dark metallic green or provide a dark metallic sheen under acidic conditions. So a dark metallic sheen means an increase in acid, which means a decrease in pH, which is the result of fermentation. So let's look here on the plate. First thing you notice is that nice dark metallic green sheen. That's an indication that there is a lot of acid produced. That means that due to the fermentation of lactose, this particular organism decreased the pH quite significantly in the plate. E. coli is a member of the enterics that has the ability to produce lots of acid when it's fermenting lactose. Therefore, E. coli gives a dark metallic green sheen on a plate. We can also see here a slight color change. We could see that's a pink color. Pink is an indication of some acid or little acid produced. So what that means is fermentation occurred, acid was produced, but we haven't lowered the pH to the extent that E. coli did. For example, the organism Enterobacter, another member of the enterics, has the ability to ferment lactose without producing much acid. Therefore, Enterobacter gives a nice pink color on an EMB plate. Lastly, here, we can see that those organisms are colorless. That particular organism actually takes the color of the plate, if anything. What that means is we had no fermentation. There was no acid produced. Therefore, there was no color change. Examples of members of enteric that would give that result would be Salmonella, would be one example. Shigella would be another example. So what we can see here is, first of all, if we obtained a clinical sample, we've isolated only the gram-negative bacilli. So those gram-positives were inhibited. So the only thing that would grow on the plate are the gram-negatives. Now, when they do grow, how can you distinguish amongst the gram-negative bacilli? It's the ability to ferment lactose that allows us to observe differential patterns of growth. In other words, different colors on the plate. Those that produce a lot of acid in fermentation of lactose turn a nice dark metallic green sheen. Those that can ferment lactose with little acid turn pink, and those that do not have the ability to ferment lactose do not change color. In other words, they're colorless. So let's move on now to the third type of selective and differential media, which is McConkey agar. Now, we did not use this one in the laboratory, but it is a very common selective and differential media. The selective agents are biosalts and a dye called crystal violet. It is these biosalts and dyes that inhibit gram-positive organisms, i.e. gram-positive cocci, while allowing for those gram-negative bacilli to grow. So what we see here on a plate is we see the growth of 3 different organisms, all of which must be gram-negative bacilli. So now, how can we differentiate, how can we distinguish amongst members of the gram-negative enterics? We do so based, again, on their ability to ferment lactose. As we talked about before, lactose is a disaccharide that can be fermented by particular members of the family of enterics. Under acidic conditions, there is a pH indicator within the plate that will turn red under acidic conditions. At a pH lower than 6.8, the media turns reddish. Another way to think about it is it turns sort of reddish pink, as we see there on the right. Now, a pH greater than 6.8, essentially neutral pH, the cells remain colorless. So this is a way for us to distinguish based on their ability to produce acids using the process called fermentation. So let's look here at the plate. What we see here on the left, we would call that lactose negative. What that means is that organism was unable to ferment lactose, therefore the cells remain colorless. The members of enteric that would give that result would again be Salmonella and Shigella, for example. Now what we can see in the middle there is lactose fermentation in organisms 2 and organism 3. We can see that the color on the plate has changed. The color has gone towards a pink/red color. Therefore, we call those organisms lactose positive, which means that they fermented lactose. So the ability to ferment lactose caused the color to change on a plate. Examples of the members of enterics that would give this result include E. coli, Enterobacter, Klebsiella, for example, would provide that nice red color. So what you can see here, again, is out of a clinical sample, we've only isolated gram-negative bacilli, and then we've differentiated based on their ability to ferment lactose. That is what a selective and differential plate allows us to do.